All right, in chemical engineering, we're all about building things, right? Usually with steel, pipes, giant reactors. But what if? What if our next factory wasn't a building at all? That's what we're digging into today, this incredible frontier where we're literally building with biology. Think about your day-to-day. You're dealing with reactors, catalysts, constantly worrying about yield. Okay, now picture all of that, the entire chemical plant, the whole shebang, shrunk down and happening inside one single living cell. Wild, right? And that right there, that's the core idea. We're not just, watching biology under a microscope anymore. We're actually treating the cell like a tiny programmable factory. We can go in, give it new genetic blueprints, retool its assembly lines, and get it to crank out the exact chemicals we want. So there's a name for this, of course. This brings us to metabolic engineering. And it's all about that key phrase, purposeful modification. It's about getting in the driver's seat, taking control of all those chemical reactions happening inside the cell to get a very specific outcome. Could be a new biofuel, could be a life-saving drug, you name it.
Okay, so how on earth do we actually do this? Well, it really boils down to a classic two-step engineering loop. First up is analysis. We have to map out and measure the flow of molecules inside the cell. That's called metabolic flux. Basically, we figure out what's going where. And once we know that, we move to step 2, synthesis. This is where we use amazing tools like CRISPR to go in and make surgical changes to the cell's DNA, redirecting that flow. So it's analyze, then act.
And this two-step process, it represents a huge, and I mean huge, paradigm shift. We're moving away from just crossing our fingers and leaving biology to chance. Now it's all about intentional, rational design. The old way, man, it was what we call a black box approach. You'd literally blast cells with radiation or chemicals and just hope that one of them randomly mutated in a way that produced more of what you wanted. You had zero idea what actually changed inside. But today, it's a white box. We can pop the hood, see the internal wiring diagram, model the whole system, and then go in and make super precise, targeted changes. This table really lays it all out, doesn't it? On one side, you've got the old way, random mutations, a total black box. On the other, the modern approach, with targeted tools like CRISPR. It's the difference between being slow and relying on pure luck, and being fast, predictable, and scalable. And what's driving this? You've got amazing advances like genomics, letting us read the cell's entire blueprint, and even AI helping us model and predict the absolute best changes to make. Okay, that's the theory. But what does this look like in the real world? Let's check out a classic case study that really shows this in action. The production of a polymer building block called PDO. So here was the problem engineers were facing. There wasn't a single microbe out there in nature that could do the job perfectly. You had some that could make PDO from one chemical, and then you had others, like good old E. coli, that could chow down on cheap sugar, but they couldn't make the final product, a classic engineering puzzle. And the solution? It was absolutely brilliant. Instead of just hoping for some random mutation, engineers used rational design. They went gene hunting. They identified the exact genes for the chemical steps they needed from totally different organisms, some from yeast, some from other bacteria, and then they literally stitched those genetic instructions right into the E. coli genome. And the result, a totally new synthetic metabolic pathway. They called it a chimeric pathway because it was a mix of different organisms, and it did something nature couldn't do on its own. It efficiently turned cheap glucose directly into a high-value industrial chemical. Now think about that. This wasn't just a small improvement. It was the creation of a biological process that had never existed before. That, right there, is the power of metabolic engineering. But you know how it is. With this incredible new power comes a whole new set of engineering challenges. This stuff is amazing, but it's definitely not a solved problem. I mean, think about it. We're asking these tiny cells to do a ton of extra work, right? That creates what we call metabolic burden. It just drains the cell's energy, and what's really wild is that over time, the cells can actually evolve to just shut down our engineered pathway to save their own skin. And if that's not enough, sometimes the very thing we're trying to make can end up being toxic to the cell that's making it. Tricky stuff. And it's not just about the biology. Beyond those hurdles, you've got the real-world rules of the game. We need super strict biosafety to make sure these modified organisms stay contained. We've got really complex legal questions, like who actually owns a synthetic DNA sequence. And to make this a real, solid engineering discipline, we need standards. We're adopting cycles like design, build, test, learn, a classic engineering loop, all in an effort to make biology just a little more predictable.
And all of this really brings us to the core challenge for the future of this field. And honestly, for you. Biology is just incredibly powerful, but it's also messy. It's unpredictable. So the big question for the next generation of chemical engineers is this: how do we take a living, breathing, evolving system and engineer it to be just as reliable as the massive plants we build out of concrete and steel?